We're going to cover the brain. But before we get started talking about the brain, I want to tell you a little bit how the nervous system developed. So if you look at the embryonic development of the nervous system, it looks something like this. Now, of course, there's a whole lot more going on, but along the dorsal side of the embryo, some things begin to happen. Around day 19, there's an area in the very center running down the midline that begins to grow and thicken. And so neurons are forming. This area is called the neural plate. If you watch the neural plate, it continues to grow and it begins to fold in on itself. So by day 20, it's formed a groove. By day 22, the ends are almost touching each other, and so the groove is very deep. And by 26 days, what's happened is it's folded into and formed a tube. This is called the neural tube. And look, it's hollow. At the same time that this folding occurs, some parts of this begin to pinch off. These are called neural crests. And so the neural crests become completely separated from the neural tube. Those neural crests are going to form the sensory neurons of the body. And so their cell bodies are going to exist outside of this tube. The tube, on the other hand, forms the brain and the spinal cord, and then all of the motor nerves associated with that. Well, if you continue to watch the development, in the beginning, this tube looks exactly the same all the way throughout. You cannot tell one end from the other. But a little bit later on, one end begins to grow faster than the other. That end is going to become the brain. And the first thing that happens is that area sort of divides into three different regions. We call these the forebrain, midbrain, and hindbrain. Now, technically, they're called prosencephalon, mesencephalon, and rhomencephalon. But for now, let's just use forebrain, midbrain, and hindbrain. If you continue to watch the development, they continue to grow. And then what happens is these three swellings, which are called primary brain vesicles, they divide, and we go from 3 to 5. So we have 5 secondary brain vesicles. So our forebrain splits, and we get 2 parts. One part is called the telencephalon, another part is called the diencephalon. So together, even as an adult, these make up the forebrain. The midbrain doesn't change very much, and it doesn't divide. And so it was called the mesencephalon. While it was a primary brain vesicle, it's still called the mesencephalon. If you look at the hindbrain splits similarly to the way the forebrain does. And when it splits, we form 2 secondary brain vesicles, the metencephalon and myelencephalon. Well, you may never have heard of these words, but we're going to use these words throughout to designate different regions of the brain. And we're going to look at the structures that form these different regions. And so if you look at the telencephalon, what we're going to see is the cerebrum. If you look at the diencephalon, it's still called the diencephalon. If you look at the mesencephalon, most of the time it's called the midbrain. If you look at the metencephalon, we're going to have 2 structures, which are the pons and the cerebellum. And then if you look at the myelencephalon, it forms the medulla. The rest of this tube forms the spinal cord. So here are our secondary brain vesicles. And again, these are going to become adult brain regions. And so when you look at your checklist for lab, this is how it's divided up. Well, if we were to use this on a normal PowerPoint, you would see these colors pop up one at a time. But this is an adult brain. And so you can still see these different regions. So the upper region, which is colored yellow, that is that telencephalon. And the center is this diencephalon. Again, those 2 make up the forebrain. And then the next thing is this middle section, which is the midbrain or mesencephalon. And then we have the lower parts, the hindbrain, the metencephalon, and myelencephalon. And again, here are those adult structures that are going to form these different regions. Well, if we look at the adult brain, it does not give us a very good indication of what the brain in the embryo and the fetus looks like. And so if we look at the development, and you can see the changes all the way through. So in this picture, you can see at 5 weeks. So you should be able to pick out telencephalon, diencephalon, and so on. And they continue to change and develop. Well, they kept the colors the same so that it's easy for you to follow through. So from 5 weeks to 13 weeks, look what has happened to the telencephalon. It's grown tremendously. Now the other areas grow too, but not nearly at the rate that the telencephalon does. If you go from 13 to 26 weeks, what you're going to begin to see then is the outside of the telencephalon begins to fold in on itself. It begins to wrinkle. The reason for this is to increase the surface area because the number of neurons is increasing rapidly. And then you go from 26 weeks to birth, the brain looks pretty much at birth the same way it does in an adult. And you can see those different regions. Well, the telencephalon is so big that it covers up parts of the other regions. And so in order to see those, you would actually have to dissect the brain in half. But in this picture, you can see through the telencephalon to find the diencephalon, the mesencephalon, the metencephalon, and the myelencephalon. Another thing about the nervous system is it has the same basic pattern throughout. So remember, it started out as this tube, and it was all one tube. And then areas of the tube enlarged and so on. Remember, the tube is hollow, and that hollow space remains throughout a person's life. And so if we look at this pattern, if we start with the spinal cord looks like this down at the bottom of this picture. Look, there's a central cavity. It's hollow in the middle. And then surrounding it is this butterfly-shaped gray matter, which we talked about in a different video. And then around that is white matter, which we also talked about in that video. So the basic pattern here is hollow, gray matter around that, and white matter on the outside. If we move up to the brain, the pattern is similar, but what happens is we add additional areas of gray matter. And when you add more gray matter, that means more cell bodies. And so if we look at this and move up to the brain stem, again, it's hollow in the middle. There's a central cavity. There's gray matter around that, and there's white matter around the outside. But notice we've added more gray matter, sort of like islands inside the white matter. But the pattern is still basically the same. We just have additional gray matter. If we move all the way up to the top part, again, we're going to see the same basic pattern. Hollow in the middle, gray matter around that, white matter on the outside, and then we have these islands of gray matter just like before. But now we've added a whole new layer of gray matter. It's called the cortex. And so this whole new layer, this cortex, is additional gray matter, additional neurons. If we look at the brain, this is the superior view, we can see that gray matter on the surface. So all of this that you see, that's gray matter. And look how folded it is, almost like an accordion. Those folds, again, increase the surface area. If you look at it, it looks like a sphere. And so it's called the sphere or the cerebrum. The word cerebrum means brain. But look, it's divided into two half spheres. And a half sphere is called a hemisphere. What divides it in half is a deep fissure called the longitudinal fissure. Now on the right side of this brain, they have removed the membranes, so you can't see the Dura Mater or the arachnoid. The Pia Mater you can't really remove. But on the left side, The arachnoid is still there. They also left the blood vessels that you can see covered or just beneath this arachnoid. If we look at it in a lateral view, it looks like this. Again, we can see lots and lots and lots of these folds. And you can see just how big the cerebrum is based on the other structures. Now, the other structures, some of them are hidden, but we can still see the metencephalon and myelencephalon. Remember, the metencephalon is the pons and the cerebellum, and the myelencephalon is the medulla oblongata. They've left some blood vessels on here so you can see just how many blood vessels there are. Remember that this whole structure is hollow. Started out hollow, it remains hollow throughout your life. Some hollow spaces are large, some are smaller. The larger hollow spaces are called ventricles, and there are 4 ventricles. The ventricles look like this. Here are those hollow spaces. There are 2 large ventricles in the telencephalon, and then there's a 3rd one in the diencephalon, and the 4th one is down near the met and myelencephalon. Well, again, this is just a few weeks. If we look at it in the adult, it looks like this. But again, you can still see these 4 ventricles. 2 of them are C-shaped, and they're called lateral ventricles. So there's a lateral ventricle inside each cerebral hemisphere. So we have a left lateral ventricle and a right lateral ventricle. They are connected to the ventricle in the diencephalon, which is called the 3rd ventricle. And then the 4th ventricle is down at the met and myelencephalon. Well, you can also see that they're connected to each other, and the connections are narrow tube-like structures. There are connections between the lateral ventricles and the 3rd ventricle. These are called the interventricular foramina. And there's a connection between the 3rd ventricle and the 4th ventricle. That's called the cerebral aqueduct. And then finally, the 4th ventricle is connected to the hollow space inside the spinal cord, the central canal. Well, if we were to fill these up with latex or plastic or something and then remove the surrounding tissues, it would look something like this. And if you notice, it looks a lot like a rooster. So you can see the rooster has the combs up there at the top. It has a face with even an eye. It has sort of a beak-like structure in the front, and then a long skinny neck, and then maybe shoulders. So those are the ventricles and their connections. We talked about the protections of the spinal cord. Well, the brain has those same protections. So first we have hair, and then we have the scalp. Then we have the skull bones, but there are also these same membranes around the brain that were around the spinal cord. Remember, they're called meninges. If they're around the spinal cord, we call them spinal meninges. If they're around the brain, they're called cranial meninges. But they're the same 3 layers. So we have a Dura Mater, we have an arachnoid, and we have a Pia Mater. If we look at the Dura Mater, remember it's this thick, tough, leathery-like structure. It's made out of dense, irregular connective tissue. It completely surrounds the brain, but it also does something else. It folds in, and where it folds in, it anchors and stabilizes the brain. It attaches to the skull. And so it's a little bit like seat belts that you would have in your car. And think about why you have seat belts anyway. A seat belt, what it does is it keeps you from moving if you get in a crash. It keeps you from flying forward and hitting your steering wheel or hitting the dash or hitting the windscreen or something like that. In other words, it keeps your car from hurting you. And that's exactly what these folds do here. There are 3 of these. And what they do is they keep the brain from moving around inside the skull. One of them is called the falx cerebri, one is called the tentorium cerebelli, and then the third one is the falx cerebelli. This is what they look like. And so again, remember, it's dense, irregular connective tissue. It's just like the dura, and it folds in and anchors to the brain. This is a GIF when you look at it on the PowerPoint, and what it is when this person strikes this skull, the brain moves back and forth inside the skull cavity. And if you think about that, every time the brain did that, it would bang into the skull on the front, it would bang into the skull on the back, and so on. And so what would happen is that the skull would be bruised every time that it hit the skull. The brain would be bruised. And so if you anchor it, if you hold it in place and don't let it move around, then that doesn't happen. That's what these folds are all about. And remember, a good analogy for these is seat belts. They're holding it in place, not allowing it to move. And so if something hits you in the front of the head, your brain doesn't go moving inside your skull. If something hits you behind the head, the brain is stabilized and doesn't move. Young children do not have very well developed dural folds. And so young children, infants, if you do the same thing to them that you do to an adult, their brain does move around a lot more. And you can get stuff or problems like shaken baby syndrome. So see if you can try to remember those names. So again, we had the falx cerebri, the tentorium cerebelli, and the falx cerebelli. So see if you can put the right name with the right fold. That's the Dura Mater. We also have the arachnoid. And again, very similar to around the spinal cord. Arachnoid is this middle layer. It has these fibers that make it look a little bit like a spider web. No blood vessels. But what it does have here that it doesn't have around the spinal cord is it has little tiny protrusions. These little protrusions are called arachnoid villi. And the best way to describe an arachnoid villus is to think of it like a drain, like you have in your sink in your bathroom or your kitchen. Because what it does is it drains cerebrospinal fluid. We haven't talked much about cerebrospinal fluid yet, but we will. But cerebrospinal fluid is made from blood, and it circulates throughout the brain and spinal cord, both internally and externally, and then it drains back into blood. So in this picture, you can see the superior sagittal sinus. That's actually a large vein filled with blood. And you can see those little arachnoid villi, and that's where this cerebrospinal fluid drains off and into that blood in that sinus. We blow it up. Here you can see them pretty well. You can get an inflammation of the meninges due to an infection. We call that meningitis. And of course, you can have spinal meningitis, but you can also have cranial meningitis. And one of the things is if you happen to have that is you get scarring on the meninges. And that scarring can cover up and clog these arachnoid villi, which means the cerebrospinal fluid doesn't have a drain anymore. And it's just like a clog in the sink in your bathroom. There's no place for it to flow. And so what happens is cerebrospinal fluid is continued to be produced, and so pressure builds up inside the skull, and that pressure can actually crush the brain. If we look at the Pia Mater is exactly the same around the brain as it is around the spinal cord. Remember, it has lots of blood vessels. It clings very tightly to the brain. You really can't get it off. It's a very delicate connective tissue. It has lots and lots and lots of blood vessels. And in this picture, you can see little arteries and little veins run all along here. So let's look at the different regions of the brain. And today we're going to look at the Telencephalon. So remember, the Telencephalon is the largest part, and it's also the most superior part. If you look at the adult structures that are included in the telencephalon, they're the cerebrum. And the cerebrum you can divide up into the cortex, cerebral white matter, and basal or cerebral nuclei. So this is what we're going to talk about now. Remember, the cerebrum is split into two parts. So it looks like a sphere. So when you split it into two halves, they're called hemispheres. So there's a right hemisphere and a left hemisphere. The word cortex means rind, like an orange rind or a lemon rind or a watermelon rind. It means it's something on the surface. And so the cortex, you can see a little band of it around here. That's going to be gray matter. But remember, inside that we have white matter, and then scattered around we have those little islands. Those are the basal or cerebral nuclei. We're going to look at each one of these. And we're going to look at the cortex first. But the telencephalon is by far the biggest part of the brain. It's 80–83% of the brain mass. And if you look at the cortex is not flat. It's folded like an accordion. So we're going to have ridges and we're going to have grooves, just like an accordion. The ridges are called gyri. The grooves are called sulci. But in some places, the sulci are really, really deep. And in those places, they're called fissures. So here we can see gyri and sulci, and we can see one fissure, the longitudinal fissure. A good way to sort of imagine this or look at it is to think of it as a bunch of snakes or worms lying right next to each other. So the snake or the worm is the gyrus. The little space in between, the grooves between the snakes or worms, those are the sulci. Each one is a sulcus. And then you can see the longitudinal fissure is a very deep groove. If we look at it from the side, again, we can see these gyri and sulci. We can also see another fissure, although it's not labeled in this picture. There's a fissure between the cerebellum and the cerebrum. That one's called the transverse fissure, but again, it's a deep, deep groove. Well, when we look at the cortex, remember this is gray matter. And gray matter, remember, is cell bodies, synapses, dendrites. It's where EPSPs happen and IPSPs happen. It's where presynaptic facilitation and presynaptic inhibition happens. It's where those neurons are arranged in circuits, all those things that we've talked about before. And so all kinds of things are going on in the cerebral cortex. This is where your memories are. It's where main thought processes are happening. This is where we control our skeletal muscles. This is where our vision comes so we can interpret it. And this is where all those things happen at the same time. And if you look at this picture, you'll see that they are regions within the cortex. And so the cortex is sort of divided up. This area does one thing and that area does another. But all these areas are interconnected. Well, if we're going to talk about all these different areas, it makes it much easier if we can divide it up into smaller portions. And so if you look on a typical person's brain, there are certain sulci that are pretty much the same. And we're going to use those sulci as boundaries to divide the hemispheres up into lobes. And there are 5 lobes. There's a frontal lobe, a temporal lobe, a parietal lobe, occipital lobe, and one called an insula on each side. Well, if you look at these names, frontal, parietal, temporal, occipital, it should remind you of the bones of the skull. And basically, the frontal lobe of the brain is underneath the frontal bone. The parietal lobes of the brain are underneath the parietal bones, and so on. Well, the way we're going to divide it up, here are these different lobes, is we're going to have to figure out sulci, boundaries. And so there are 3 of these sulci we're going to use. One is called the central sulcus, and the central sulcus separates the frontal from the parietal lobes. Another one is the lateral sulcus, and that's going to separate the parietal from the temporal lobes. And then we have one which you can't really see from the outside, but you can if you open it up. It's the parieto-occipital sulcus, and that's going to separate the parietal from the occipital lobe. And so when we look here, you can see the central sulcus, the lateral sulcus. Again, the parieto-occipital sulcus is a little internal, and so in this view we can't see it. So, anterior to the central sulcus is the frontal lobe. Inferior to the lateral sulcus is the temporal lobe. And so you should be able to label these lobes as well as these sulci. If you take a chunk out, you can see that what we're talking about is this surface, it's the cortex. And remember, the cortex is gray matter. You can also see sulci and fissures and see the comparison between the two. So we saw frontal lobe, parietal lobe, temporal lobe, occipital lobe. We can't see this fifth lobe from the surface because it's folded in. It's folded in. If you could take a pair of separators and pull the lateral sulcus open, you would be able to see the insula. It's also gray matter. It also has sulci and gyri. It's also cortex, but it's just not visible. It's sort of insulated, and so that's why it's called the insula. But here are those sulci. So the central sulcus, lateral sulcus, parieto-occipital sulcus. And again, we're going to use those to determine these lobes. Remember, you can't see the parieto-occipital sulcus from the surface, but if you do a midsection, a midline insection, you can see the parieto-occipital sulcus, and you can see it here in this dotted blue line. And so everything posterior to that is the occipital lobe. Well, since you can't see it from the surface, you sort of have to imagine where it is by looking at it from the inside. And so in this dotted blue line, you can see it is here as well. And so again, here are our different lobes. So take a minute and decide what these lobes are and what these colored lines mean and so on. So we're talking about the cortex. Remember, the word cortex means rind. And think about a lemon rind or an orange rind. It's not very thick. And that's exactly the way the cerebral cortex is as well. It's only about 1/16 of an inch thick. But remember, it's folded like an accordion. And so because it's so folded up, there's so many of these gyri and sulci that if you were to measure how much of it is, it's about 40% of the cortex. It's about 40% of that brain mass. And the cortex is where all of the things that make you a person, this is where it's located. This is where your memories are. It's where your personality is. It's where you make decisions and communicate. It's where you tell your body to move. All those things are located in the cortex. And so if we look at this cortex, different areas are responsible for different things. So in this picture, you can see the cortex. So this is a frontal section through here. And you can see the cortex is the gray matter on the outside. And again, you can see these gyri and sulci. If you look where it says cerebral cortex on the left and see how folded in that gray matter is right there, that's the insula. So if you look on the insula, you can see it on both sides. It's this deep gray matter. Now it's still surface, it's just folded in. So it's still part of the cortex. Also in this picture though, you can see white matter. And if you look down at the bottom of the white matter, you'll see these gray, almost shaded areas. Those are the cerebral nuclei. Well, we're going to talk about all those things, but for now, let's look at the cerebral cortex and what it does. So here's sort of a just general thing about what each area does. And so you get some idea that there's sort of a map on the brain of the things that it does. Well, these areas are called functional areas. They're also called Brodmann's areas. Brodmann was a Russian scientist, and he was one of the very first people to begin to map out the brain. And he did it in a time when there wasn't a lot of equipment. And so what he did, he used histological slides. And what he found out was that there are layers of neurons in the cortex. And those layers are different in different areas. And so he was able to build sort of a map of the brain. And it turns out that there are 3 different types of these Brodmann's areas. Their names describe what they do. So there are motor areas. Motor areas control voluntary movements. In other words, they send their axons down to all of your muscles. So when you decide to lift your arm or to move your hand or to smile or anything like that, it starts in one of these motor areas. There are also sensory areas. So sensory areas are going to receive information from all of your senses. And so the information is going to come in from your skin, it's going to come in from your eyes and your ears and so on. And what happens here is we become consciously aware of that sensation. In other words, we realize that something is touching us, that we're seeing light, that we're hearing a sound. And then we have these areas called association areas. And what association areas do is they take that information that just arrived and they figure out what it means. They integrate it. They compare it to your memories and they figure out what it is you're looking at, what's touching you on the skin, what does that sound mean. And so we're going to look at some of these areas. Here's a map of the functional areas. And look, every color means a different area. Every number means a different area. Well, if you look at this, there's 45 different or 47 different areas on here. We're not going to cover anywhere near that many. We're just going to look at a few of each of the different types. So we're going to look at motor areas first. And we're only going to look at 4 motor areas. One of them is called the primary motor area, sometimes the primary somatic motor area. Another one's called the premotor area or premotor cortex. And then we have Broca's area and we have the frontal eye field. Well, you should be able to find all of these on here, but we're going to look at the primary motor area, and that's this bright red one labeled number 4. And so when we look at it, you can see it here. Remember, we use the central sulcus to divide things up. Well, the primary motor area is located right next to the central sulcus. In this picture, it's green, so it's in the frontal lobe. It's just anterior to the central sulcus. It's a little different than most of these other areas. Most areas encompass several different gyri, but the primary motor area is located entirely on one gyrus, the gyrus just anterior to the central sulcus, and so that gyrus is called the precentral gyrus. So on that precentral gyrus, which we can see right here, is this primary motor area. And we can see it here as well. We go back, find the central sulcus, and then there's the precentral gyrus. Now, it's not as easy on a human brain, a whole brain, as it is on a drawing. Well, what you have on this precentral gyrus are a bunch of pyramidal cells. And those pyramidal cells are going to send their axons down, and the axons are going to make up the pyramidal tracts. And remember, the pyramidal tracts all start with cortico. So what we're going to have here are the corticospinal tracts and the corticobulbar tracts. And what they do is they go to all of our skeletal muscles. And when they go to those skeletal muscles, what they do is they allow us to have voluntary movements. They're conscious control, conscious, specific, precise, skilled control of our muscles. And if you look at this precentral gyrus and you map it out, what you find out is there's a little man, a map of a little man on this gyrus, so that each part of that little man matches the same thing on your body. It's called a motor homunculus. And the motor homunculus, homunculus just means little man, looks like this. So if we look at it, here's this single gyrus here. And so you can find all the different parts that compare to a different body part. So see, all of the muscles for the face are controlled here. All the muscles for the hand, they're controlled there, and so on. Well, if you look at this, though, you can see this little man. But look at the proportions. Do you think the proportions match a normal person's proportions? They don't. And so it doesn't have anything to do with how big your body part is. It has to do with how much motor control you have over it. Find the trunk on here, and you can see, even though it's the biggest part of our body, there's very little brain power devoted to the trunk. Because what can you do with your trunk? You can bend this way or that way, you can straighten, you can do things like that. But that's about it. But look how much brain power is devoted to your hand. Because how many different movements can you make with your fingers and your hands? Look at how much brain power is devoted to your face. Because think about this, how many different facial expressions do you think a person can make? The number's probably unlimited. And look, there's even a lot of brain power devoted just to the tongue, because we can move our tongue in lots and lots and lots of different ways. But you don't look like this. If you did, you'd look like this. So this is what that motor homunculus looks like if it were a real person. So again, it's not the size of the body part on your body, it's how much motor control. And so when you look at the motor homunculus, you can easily tell which body parts you have the most motor control over. If we compare the little man with the drawing, you can kind of see the difference here. Let's look at another one. This one's called the premotor cortex. And the premotor cortex is really just in front, just anterior to the precentral gyrus, just anterior to the primary motor area. So if we look at it on a map, it's this number 6 up here. And so it's just anterior. And what it does is it stores patterns of muscle movements. So when you learn to do something and you learn to do it well, your brain realizes that there's a pattern associated with it. And this muscle needs to fire this time, and this muscle needs to relax, and this muscle needs to fire next, and so on. Well, once you learn that pattern, you store it. So think about walking. Walking has a pattern to it. And so if you think about a baby when they first come around, even when they start standing up, they cannot walk. And then when they learn to walk, think about what it looks like when a little kid walks. It doesn't look like an adult. They are up on their tiptoes, they fall forward, move their legs really fast, fall down a lot. But every time that they do all those things, they're learning better and better ways to keep from falling down, better and better ways to move their legs. And so eventually a pattern of when the muscles should contract and when they should relax becomes established. And when that happens, we store it in this premotor area, premotor cortex. Same thing happens if you learn to play the piano. People who play the piano know where the keys are. Their fingers just seem to know. Think about typing. You don't have to look at the keyboard to type. Your fingers know where the key is. So the brain has this pattern built into it. Another thing the premotor area does is it helps to plan movements. So you can think about, I want to do a layup. I'm going to have to bounce the ball, run, and then at the same time I'm going to have to jump and move the ball up and try to push it against the goal and into the goal. Well, the first time you do that, it probably is not very good, but still you can see what it is you're supposed to do. And so the premotor area helped you plan those movements. And then as you do it more and more and more, we refine the process, we store the pattern, and so on. And so if you damage the premotor area, what happens is you lose those patterns and you forget how to do things. So even though you might have been riding a bike your entire life, you know the pattern, you can ride it with no hands, you can make it do tricks and stuff like that because your body, your brain, everything works together in that pattern. If you damage this area, you might not be able to stay up on the bike at all. Now, that doesn't mean you can't relearn it, but as you relearn it, it's just like it was the first time you learned it. At first, it's going to be very clumsy and labored until you reestablish the pattern. Let's look at Broca's area. So Broca's area, if you look at it on the map, it's number 44 here and 45. Broca's area is a little bit different from every other one that we're going to talk about, or at least most of them. And that is, it's not on both sides. If we look at the primary motor area, it's on both sides. If we look at the premotor area, it's on both sides. But Broca's area is only on one side. And about 90% of the people, it's on the left side. What Broca's area does is it directs the muscles of the tongue when we speak. So if you think about saying something really complicated, your tongue has to move in lots of movements, lots of directions. It has to push, it has to do all kinds of things in order for you to make those sounds. And so that's what Broca's area does. It controls the muscles of the tongue. It becomes very active when you're prepared to speak. And so if you were to say something like a tongue twister of some sort, think about what your tongue is having to do. It's making like crazy movements. If you say Peter Piper picked a peck of pickled peppers, if Peter Piper picked a peck of pickled peppers, where's the peck of pickled peppers Peter Piper picked? Think about all the movements my tongue made while I was doing that. That's Broca's. So if you damage Broca's, what happens is your tongue just doesn't make the movements you want it to make. So there may be certain sounds that you just can't make anymore. Think about the sound th, th, th. Everybody do it and think about what your tongue is doing when you do th. Look, it pushes against the back of your teeth and then it pops off in order for you to make that sound. Well, if you damage Broca's, you may never be able to make your tongue do that, no matter how hard you try. So th is never going to come out. It's going to come out like wuh or something. And so it makes speech very, very difficult. So here's Broca's. Remember, only on one side, usually the left. And then the last one we're going to look at is called the frontal eye field. And the frontal eye field is a really easy one to understand because its name tells you exactly what it does. It's in the frontal lobe and it controls where your eyes are looking, your eye field. And so it controls voluntary movement of the eyeballs. And so if you look at it, you can see it here. It's at number 8. That's the frontal eye field. And so if you damage this area, your eyes, one or both, don't move the way you think that they should move. So if you want to look at something out of the corner of your eye, instead of moving your eyes in that direction, you may have to turn your head all the way around just to see it. Now, if you look here, you'll see there's other motor areas as well, but we're not going to talk about those. There's our frontal eye field. So these are the motor areas that we looked at. So hopefully you'll be able to review those, know those, understand what it is that they do, where you can find them. Let's look at sensory areas. So sensory areas, remember what they do is they receive sensory information. So sensory information from anywhere in your body is going to come to your brain. And this is where you're going to become consciously aware of it. So we have a sensory area for all of our senses. Well, if you look at this list, you're going to recognize primary visual area, that's going to be about eyes. Primary auditory, that's going to be about ears. Olfactory, about nose. And gustatory is about taste. But somatosensory is your body senses. So think about if you were to plug up your ears and close your eyes and all those things associated with the head, you could still find your way around in a room. You could find your way lots of places. You could tell if things were touching you or what the size of things was and so on. Our skin has tons of sensory information. So do our muscles and joints. That's called somatosensory. So somatosensory happens everywhere on your body. Visual only happens in the eyes. Auditory only happens in the ear, and so on. So somatosensory is a much bigger sense than these others. But every single one of these has their own area. And here you can see these different areas. If you look at the size of the brain part, you get an idea how sensitive each of our senses are. So our two most sensitive senses are our body senses and vision. If you look at some other animals, they don't match up the same way. Some animals are much, much more dependent on their sense of smell, and so they have much bigger olfactory areas, and so on. But let's look at these. Here they are on here as well. And the first one we're going to look at is this primary somatosensory area. So this is another one similar to our primary motor area that's located on a single gyrus. Remember, the primary motor area was on the precentral gyrus. Well, the primary somatosensory area is on the postcentral gyrus. In other words, it's the gyrus right posterior to the central sulcus. And if we look at it, look, it runs sort of parallel to the primary motor area. In this picture, it's blue. So it's the number 1, 2, 3, that's the primary somatosensory area. Well, what it is that all the neurons from your skin and from your skeletal muscles and from your joints that are carrying sensory information, they go up your spinal cord. We talked about the dorsal columns, the fasciculus gracilis and fasciculus cuneatus. We talked about the spinothalamics, the anterior and lateral spinothalamics. All of those information, they go up. They're going to go up the spinal cord, and this is where they're going to terminate. They terminate on this postcentral gyrus, this primary somatosensory area. Remember, there's a map up there, and we talked about how things cross over. Remember, that's decussation, and then we talked about it goes to a very, very specific spot, and that's somatotopy. And so once again, if we look at it, we're going to find a map up here. And the map is going to look like a little person again. So we're going to have a different homunculus. This one is called the somatosensory homunculus. And so it's a map of the sensory areas. So if we look at that, here is that gyrus, and here is this homunculus. It looks like this. So once again, you can find all of your body parts here. But once again, when you look at it, the proportions don't match. But it doesn't have anything to do with the size of a body part. This time it has to do with how sensitive that body part is. How much sensory information can you gain from that body part? And so the bigger the body part on the map, the more brain that's devoted to it, the more sensitive the area is. Now, if you look here, you'll see genitals here. We didn't have that on the motor homunculus because we don't have much control over our genitals skeletal muscle-wise, but they are very sensitive. But again, look how sensitive things like your fingers are and your face and your tongue and so on. But as I said before, you don't look like this. If you did, you would look like this. So here is our somatosensory homunculus. And so by looking at the size here, again, you can tell how sensitive the body parts are. You might think that this one looks exactly like the other one, but it doesn't. The sensory homunculus and the motor homunculus don't look exactly alike. And hopefully you can figure out which one is which. Let's look at another one. This one's called the primary visual area. So visual is associated with vision. And so the only place these neurons are going to come from is from the retina of your eye. And so light is going to hit the retina of your eye and activate those neurons. So we're going to get action potentials that come back to this area. And so what we're going to see is some show up as light, some don't send action potentials, so it's dark. But what you do is you become consciously aware of a shape, consciously aware of colors, consciously aware of movement. But it does not tell you what you're looking at. You see it, but you yet don't know what it is. But if we look at the primary visual area, it's way to the back of the brain. It's in the occipital lobe. In this picture, and I don't have that picture, but anyway, you can see it here in the back of the brain. This is where this visual area is, and this picture is green. Let's look at the primary auditory area. So the primary auditory area, we only hear with one body part, and that's our ears. And so our inner ears have these neurons whose axons go to the brain, and they go to this area. So when you hear a sound, we monitor how loud it is. We monitor the pitch. We monitor the meter, what kind of rhythm it has, and all those things, they wind up here. But this area does not tell you what you're listening to. It doesn't tell you if you're listening to a bird singing or a hacksaw or something. You just become consciously aware of those sounds. All of that information on pitch, rhythm, loudness winds up here. But again, you just become consciously aware of the sounds. And here you can see the auditory area. And the auditory area is on the temporal lobe. And this picture is purple. Let's look at the primary olfactory area. So the primary olfactory area, we only are able to smell with one part of our body, and that's the nasal cavity. And so neurons from the nasal cavity project here. And so this is a chemical monitoring system. And so we smell things, we monitor these chemicals. But again, the chemicals may have one odor, they may have a different odor, and so on, but we don't know what we're smelling. We just become consciously aware that that smells sharp, that smells savory, that smells nasty, whatever, but we don't know what it is we're smelling. If you look at this, this is in the insula. So you can see it here. It's yellow. It's on the upper part of the insula. And then the last one of these is the primary gustatory area. Gusto means taste. You may have heard the slogan, go for the gusto. It means go for the taste is what it means. Well, again, we only have taste buds in our mouth. There's a few taste buds in our throat, but we only have taste buds basically in our mouth. And so those neurons, the axons, are going to wind up here, and they're also going to project to the insula. And again, we're going to become consciously aware of salty or sweet or sour, but we're not going to know what it is. We're not going to know if we're eating a pie or if we're eating a steak or whatever. We just become consciously aware that we're tasting. And so if you look here, you can see the gustatory cortex. Here it is, this red one. And they're very close to the olfactory because olfactory and gustatory are connected to each other. So those are our primary sensory areas. So hopefully you know what they detect, where you would find them, and so on. One thing to remember again, remember this is where we become consciously aware of something. It is not where we determine what it is that we're looking at, feeling, hearing, smelling, or tasting. That happens somewhere else. Okay, let's look at the association areas. So association areas, what they do is they take the information that's coming in and they figure out what it is that's going on. What am I looking at? What am I smelling? What am I tasting? What am I holding in my hand? And there's an association area for every sensory area. And they're all right next door. So remember, we had a primary somatosensory area. We have a somatosensory association area. So if you're holding something in your hand and you're rolling it around and you're trying to figure out what it is, all that information goes to the primary sensory area. Then the primary sensory area sends it to this association area. And the association area compares it with memory. How heavy is it? Does it rough? Is it smooth? And what happens is, as you roll it around or feel it or whatever, you start to get a mental image of what it is. And your brain goes through everything that you've ever felt or touched or held until it comes up with something very, very close to what it is that you're holding now. The same thing happens at the visual association area. So remember, from your retinas, movement, color, all of that goes to the primary area. And then it gets passed to this visual association area. And so you figure out what you're looking at. You figure out, wow, I'm looking at a clown dancing on top of a ball. And the only way that you can figure that out is it compares all of what you're looking at now with what you've seen in the past, with your memories. And this happens at every one of these. So again, if you look at these, they're very close, right next to the area they're associated with. So if you look at the back part of the brain, number 17, that's the primary visual area. But 18 and 19 is the visual association area. If you look a little below that, you'll see hearing. Hearing is number 42 or whatever, 41. And the auditory association area, number 22, and so on. And so, again, this is where all your memories are stored. This is where all those experiences are stored. So, if we look at the somatosensory association area, what we're going to do is we're going to get a complex understanding or a comprehensive understanding of what it is we're touching, what it is that's pushing against us, what are we holding in our hand. We're going to figure out size, we're going to figure out weight, we're going to figure out texture, we're going to figure out all those different things, and we're going to come up with an idea of what it is. Same thing's true for visual. We're going to interpret color, form, movement, all of that until we're going to decide what are we looking at. Same thing for hearing. So if you have a song that you heard a long time ago and you've forgotten about it totally, there's a memory of it stuck in your head somewhere. And so if you ever hear it again, what's going to happen is your brain is going to search around until it finds something that matches the rhythm, the pitch, the meter, the loudness, all of that, until it finds the song and you can identify the song. Same thing happens here for olfaction and gustation, except olfaction obviously is for smell. Am I smelling a cherry pie or am I smelling manure? Gustatory, am I drinking lemonade or is it medicine that tastes like lemonade? There are also some other association areas which do different things. And so we're going to look at these three as well. But again, association areas are complex areas with lots of connections to different parts of your brain. And so it can pull together all sorts of stuff. It's connected to all your memories. So we're going to look at these three. One of these is the prefrontal cortex. Another is the general interpretation area. And then finally, Wernicke's area. So if we look at the prefrontal cortex, this is way up in the front of the brain. And if you look at it here, it's way up here in the front part of the brain. And if you can see it here, it's like number 11 on this. It's not a huge area, but it's in the very most anterior part of the brain. And what the prefrontal area does is it allows us to make plans. It allows us to understand consequences. So you can think to yourself, right now we're supposed to be in lockdown with COVID, but I'm going to go out to the grocery store and I'm not going to wear a mask and I'm going to do this. And then you think to your head, well, if I do this and this and this and this, first of all, when I leave the house, I'm going to expose myself to a bunch of people. When I don't wear a mask, I'm not very protected, and so on. And so you can go through this whole plan of a scenario till you get to some sort of consequence of your action. That could be a good thing. I'm going to go and go and go and go until I become a doctor. Or it could be a bad thing. I'm going to do this and do this and do this until I wind up in prison. But you can do that. We don't think other animals can do that. Another thing about the prefrontal area is the very last part of your brain to mature. So in women and females, it matures around age 18 or 19. But for males, it's not until about age 25. So someone male and younger than 25, they're not very good at understanding the consequences of their actions. And so they tend to take lots of risks. And so you can see them doing all sorts of things that you would just think that that's absolutely crazy. Why would anyone do such a thing? And when they get older and look back on it, they may think it's funny, but they're also going to think in the back of their head, what in the world was I thinking? It sounds like it might be bad, but because younger people, especially younger males, are risk takers, there's a whole lot of things that are now considered good. When they took those risks, they were very, very, very crazy. Sailing off and looking for the new world was a crazy thing. They thought people were going to fall off the earth. Going to the moon was a crazy thing. Think of all of the things that could have gone wrong. So that's the prefrontal area or prefrontal cortex. Let's look at the general interpretation area. It's a good name for it because it's exactly what it is. It's mostly memory storage. So one way to think about memory is to think of it like having a bunch of filing cabinets and you put it in a file. And that's sort of the way it is, except when you have a memory of something, it includes a lot. It includes what you saw, but it also includes how you felt. It includes what you ate or drank or anything like that. And so what happens when we store that memory is we break it apart and we store the visual part in one place. We store the olfactory part in another place. We store the emotional part in another place, and so on. And what we do when we go to recall a memory is we reassemble all of those parts. It's a little bit like a jigsaw puzzle. And think about this. If you keep playing with a jigsaw puzzle over and over and over again, at some point you're probably going to begin to lose pieces. They're going to get stuck in your couch, or the dog's going to eat one or something. And so look, you're going to wind up with a jigsaw puzzle that's not complete anymore. But you can fill the blank spaces. We can look at a picture and think, what's supposed to go there? And you can fill it in. And that's what we do with memories too. Except sometimes when we fill it in those blank spaces, we put the wrong thing in there. And so memory is not perfect. It's not perfect at all. Another thing that happens in this area is we interpret things that are going on around us. So we take all of those memories, we use them to figure out what's happening right now. That's what the general interpretation area is about. And it's a big area. This general interpretation area is most of this purple that you see. And then the last one we're going to talk about is called Wernicke's area. Wernicke is the person's name, and he's German, so it's not Wernicke, it's Wernicke. But Wernicke's area is another one that's a little bit weird. Remember we talked about Broca's hair area and how it was only on one side? That's exactly the way Wernicke's area is. And Wernicke's and Broca's are always on the exact same side. So remember, 90% of the time Broca's was on the left. That means 90% of the time Wernicke's on the left. They're always on the same side. Remember, Broca's had to do with moving your tongue around and make sounds and so on when you wanted to talk. Well, Wernicke's is all about figuring out what you want to say. It's all about language comprehension. And so what it does is it connects things to images in your brain. It can connect a picture or a symbol or a sound or a word or whatever to a brain image. And that's basically what language is anyway. It's when we've agreed that this picture means this sound means this. And so if you look at that, this is what Wernicke's does. It allows us to make those connections. And so if I were to show you a picture, well, here you can see Wernicke's. It's this dotted line right here, number 22 area. If I were to show you a picture, you would put together in your head, it may not even be a perfect picture, but you would come up with something that you think that it means. And so if I showed you an octagon with a stick on it, and I just showed you that, you'd probably think, oh, I meant a stop sign. But if you think about the word stop, what does that mean? Stop, it's just a sound, but we've assigned something to that word. Same thing is true for all language. And so if we were to look at anything, we are going to be able to figure that out. And so if I show you this, when you look at it, you might think this is a sheep. But look at it, it's not a very good sheep. It doesn't really look like a sheep at all. But there are certain things about it that make us think it's a sheep. It looks sort of fluffy. It has 4 of those weird things. It could be legs and so on. But if you damage Wernicke's and you look at this, you're not going to see a sheep. What you're going to see is just a bunch of lines. It's not going to connect and make that sound. And then if you think about the word sheep, why even do we even call it sheep? And look at all these words. Every single one of these is a different language that means that same animal. So if you say these words to somebody who doesn't speak the language, they're going to have no idea what you're talking about. But if you tell them, das Schaf means sheep, and they remember it, the next time you say das Schaf, they're going to think of a sheep. So Ventricles helps us to do that. It helps us to connect those words, those symbols, those sounds to the correct brain image. So if you damage Ventricles, language comprehension is damaged. And if you listen to someone who has Ventricles damaged and you listen to them talking, for the most part what they say is going to sound like gibberish. But to them, they're saying exactly what they think they want to say, but the connection is messed up. This is a little video that you can watch, and it's on YouTube. It compares what happens if you have Broca's damage versus Ventricles damage. Aphasia is the inability to speak, basically, or speak correctly. So if we review those, you get an idea of all of those that we've talked about. So those are the different parts of the cortex. Well, when you look at it, I don't know who said it, but someone said once that we only use about 10% of our brain. Well, that is just total bunk. We use all of our brain, all of our brain. Maybe what they meant is we only reach 10% of our capacity, and maybe that's a little closer to being true. But we definitely use all of our brain all the time. So if we look at this cortex, we talked about these different regions, and remember most of the time when you have it on one side, you have it on the other. So we had a primary motor area on one side, we have it on the other. We have a primary visual area, we have it on the other. But there were a few times when we said that what was on one side was not on the other side. Remember, Broca's and Ventricles are only on one side, usually the left. So that means that even though the two sides of the brain are similar, they're not exactly alike. And they're different in other ways too. They process information differently. And if you look at this graphic, you get an idea of how they process information. So if you look at the left side of the brain, what you see is this shades of gray, lots of people in these little boxes, their heads are down, they're working, they're very industrious, they're doing all kinds of things. They're not talking to each other though. Look, they're isolated from each other. Everybody's doing their own little thing. The left side of the brain does something called serial processing, which is similar to what happens in the spinal cord. So the left side of the brain is very good at doing if-then and doing analytics and math and things like that, where there are steps that one has right after the other. If you think about working a math problem, you do it in steps. If you've ever taken math, a lot of times the teacher will put on the test, show your work, because they want to see if you're following those steps. That's a very left brain kind of thing. But if you look at the right brain, look at it. You see a lot of colors. There are people there, but look, the people are not doing the same kinds of things. They're not in little boxes. They're all out to thing. They're working with each other, and they're doing things that are artsy, and they're doing things that have to do with music and things like that. It's very much not regimented, it's more free. And the processing here is more parallel. And if you remember, we talked about parallel processing, and I used the example of if I said the word apple, remember you could come up with your mother, but with the same thing, when I said apple, you could come up with Ariana Grande. So in other words, one thing can lead to another that has nothing to do with the previous thing. And so the right brain and the left brain process information very differently. But notice also there's a bridge that connects these two. And so the two sides are in constant communication with each other. Well, here are some left and right brain characteristics. So left brain, very, very detail oriented. This is where our speech centers are for almost everyone. This is where your IQ is. It's where we do things like calculations and conscious actions. It tends to be very positive emotions. Whereas if you look at the right brain, it's not small details, it's big picture. It can't talk. There are no speech centers on the right for most people. And so the communication has to be nonverbal. This is not where your IQ is, it's where your EQ is, your emotional intellect. And it's not where we do math calculations, it's where we do the beauty of math, the math comprehension. It's also where social skills are and things like spatial awareness are located. So if we go back and look at that picture we were looking at before and look at all of the details on here, you get a good idea of how the right and left brain express themselves. And if you wanted to put it in one word, you could probably say the left brain is academic. And if you wanted to say what the right brain is, you might say it's creative. But again, don't forget, there's a bridge. Bridges connect these two sides. So even the creative, very creative person can be academic, and a very academic person can be creative. And sometimes we use one side of the brain more than we use the other. And it switches off. And so I'm going to ask you to do a couple of things to find out, are you using your left brain right now, or are you using your right brain right now? Remember, left brain, academic, and you can see these things that are going on. Right brain, creative, and you can see these things that are going on. Well, at this one moment, one side of your brain is probably in more control of your body and what's going on than the other one is. And so we're going to take a little test, and we're going to see right now which side is dominant. And what we're going to do is we're going to do it. We're going to take the following test. And I actually want you to do it sitting at your desk. So do these things as I tell you to do them. The first thing we're going to do is I'm going to ask you to look at this, and then I'm going to ask you to say the color as you go through here. You have to do it out loud. You have to say it. Don't read the word. Just say the color. Go. Well, how far did you get? How far did you get before you started saying the word? Well, the first thing I do when I say, look at this, is I say the word yellow. But that's not what I wanted you to do. I wanted you to say green, red, blue, yellow, blue. After a little while, though, most people will switch off, and you don't make it all the way through. And you start feeling weird because you almost feel anxious inside when you're about to screw up. There's this conflict going on between the left brain and the right brain. Your right brain wants to say the color, but remember, the right brain can't talk. So it has to force the left brain to say those words. And then the left brain gets tired of it because it wants to read the words. So you don't get very far. The next thing I'm going to ask you to do is I want you to cross your arms. Just everybody cross your arms like this. And look down and see which arm is on top. Whichever arm is on top, the opposite side of your brain is in control right at this minute. And now I'm going to ask you to do something else. I'm going to ask you to try to do it the other way. See if you can cross your arms the other way. It's probably going to feel very, very weird. By the same token, cross your thumbs and see which one is on top. And then remember, the one on top, the other side of your brain is controlling this right now. Well, is that the same as this? Is the same part on top? Well, if it's not, you're using a different part of your brain when you do those two different things. Here's another thing. What I'm going to show you is a picture. And what I want you to do is stare straight at this person's nose, and I want you to decide, is this person happy or sad? So here's the picture right here. Look at his nose. Just look at his nose and decide, is he happy or sad? Well, when you decide that, it means that one side of your brain is more in control than the other one. Because look at this person's face. One side of his face is happy, but the other side of his face is frowning. He's sad. So whichever one you decided, that's the side you were paying more attention to, and that's the part of your brain that was making the decision. Let's go back here and look at this real quick. So here's our thumb thing. The next thing I'm going to show you, what I want you to do is look at this logo, and I want you to find the hidden arrow. And let's see how long it takes you to find the hidden arrow. Well, if you're right-brained, you'll find the arrow. But if your left brain is in control, you're going to read the words FedEx Express, FedEx Express, FedEx Express over and over and over while you're looking for this arrow. Everybody find the arrow yet? Here, let me show you this. Find the E. Can you find the E? Once you find the E here, let's go back and find the arrow. Now, can you find the arrow? It's between the E and the X. Exactly. So once again, depending on which part of your brain is in control, it's going to be easier or more difficult for you to do these things. Now there's a lot of disagreement about whether people are left brain or right brain. You might hear that I'm a right brain person, I'm a left brain person. Well, nobody is right or left brained. We use all of our brain, but remember, there are times when one is in control more so than the other one is. There's a lot of conflict about that. But what is not a lot of conflict about is that males and females have different brains. So you may have heard the saying, men are from Mars and women are from Venus. That was the title of a book. Well, I don't know about that, but they are very different brains. So if you look at them, let's look at the male brain first. So the male brain forms because of testosterone. So in utero, this little fetus has testes, and those testes begin to release testosterone. And they go and they change a lot of things about the fetus, but they change the brain of the fetus. And so an adult male brain is different from an adult female brain. Males tend to be more analytical and more logical. They tend to be less emotional and less empathetic. They're also more territorial and they have shorter fuses. They're also more spatial, which means they're less likely to get lost. In fact, we even have the stereotype that a male will never stop and ask for directions. Well, there's a good reason that males actually don't need to stop and think about directions or ask for directions. If you think about way back when we all lived in one little spot and males went out hunting and gathering and all that, if they didn't know where they were and they didn't know the way back home and they got lost, they were going to die. And probably not only would they die, but the people waiting on them back at the village would die too. So males that could not find their way around were weeded out. Another thing about males is they tend to have a lot more addictions. about 10 times more addictions than females. And addictions can be all kinds of addictions: drug addictions, sports addictions, sex addictions, all kinds of addictions, gambling, all kinds of things. Males are much, much more prone to addictions than females. And then males tend to be a lot less monogamous as well. If you look at the female brain is also different. And the reason it's different is because there is no testosterone. So when the female is a fetus, there are no testes. Since there are no testes, there are no testosterone. So it develops in the absence of testosterone. And you get a different kind of brain. And so the adult female brain is very different. We already know it matures earlier. Even the prefrontal cortex, the last part to mature, matures 5 or 6 years earlier than a male. Females are much, much more vocal, and they're better at working into groups. They're better at telling each other their problems because they're empathetic, and they're better listeners than males are. They're also really, really, really good at making memories. So when a female makes a memory, they include a bunch of different things. So if you ask a male and a female about the same event in their life, like the day that they proposed and they were decided to get married. Well, if you ask them, the female, she'll say, "Oh, it was on a Sunday, and we went to the park, and there was a band playing, and we had a picnic, and I was wearing this, and he was wearing that, and we sat down on the blanket. It was so romantic, and he hid the ring in I don't know, this, and so on. He got down on one knee." So all of this detail. And then you turn around and ask the male about it, and most males would say, "Yeah, I asked her to get married, and she said yes." Now, it doesn't mean that they cannot remember those things. It's just that the male brain doesn't find all that detail that important. And then think about this. What if you go to a sporting event? You get completely opposite kinds of things. So males are good at like batting statistics and stuff like that because they're a lot more left brain. But male and female brains are very, very different. This is kind of fun to do. These are some things. The brain can be easily tricked, and it can also get very bored very easily. So one thing you can do, and you can do this at home, is if you take a ping pong ball or something like that, cut it in half and tape it over both your eyes, and then you go lay down in a darkened room, and you take a radio and turn it to a station where there's only static, and then you just lay there, and there you just hear the static so that you can't hear any other sounds. The room is dark, so there's not a lot of stimulation. What's going to happen is within a few minutes, your brain is going to start to hallucinate. You're going to have these bizarre sensory distortions. People see things like prancing dogs and horses, and they hear dead people talking to them and all kinds of stuff. Your brain gets bored, and so it invents all sorts of things on its own. Another thing is, if you ever hurt yourself, if you can make that body part far away from you, it hurts less. And one easy way to do this is with a pair of binoculars. So if you take a pair of binoculars and you look at it where it looks normal or big, and you stick your hurt finger in front of it, that finger is going to hurt like heck. But if you turn the binoculars around so that everything looks far away, and you hold your finger up in front of it, your finger will quit hurting. And then another fun thing to do, and we used to do this in our lab, is that if you do this, and I'm going to show you a little video or where you can find it, the same thing, but if you take a rubber hand and you hide the person's real hand. And what you do is you stroke their hand and you stroke the rubber hand at exactly the same time. Eventually your brain is going to connect those two because it feels this thing going on at the same time and your eye sees it at the same time. And so that rubber hand is going to feel like your own real hand. And what's going to happen is if someone takes a hammer and smashes that rubber hand, you're going to feel that pain and anxiety because your brain is convinced that that rubber hand is your real hand. Here's the link to that little video. So if you click on this link, you'll get to watch this little hand experiment. Okay, so let's review just a little bit. Remember, we said that even though we have comparative regions on each side, they're not exactly the same. The two sides are very, very different. And then we looked at the way the processing happens on the left, the way the processing happens on the right. So that was the cortex, but we also have white matter. It's called cerebral white matter. Remember, white matter is white because of myelin. The only part of a neuron that has myelin on it is the axon. So all cerebral white matter is bundles of axons. Tracts is what they are. And they're connecting the different areas of the brain. And so they allow the different areas to talk to each other. Well, when you look at it, here you can see three different types of white matter. Again, they're all tracts. They're bundles of axons, but they're running in different directions. We have commissures, we have association fibers, and we have projection fibers. So when you look at this, here's a frontal section of the brain, and look at all the white matter that's in there. And so that white matter is connecting the gray matter in different areas. Again, it's just wires running from here to there. It's areas of communication. Again, there's three types of this. One type, commissures, connect the two hemispheres. So you can see them here in this red color. We have one huge bridge, which is called the corpus callosum. And then there's a much smaller bridge called the anterior commissure. But what they do is they allow the two sides of the brain to talk to each other. And so that graphic where we had the right and left brain, remember there were bridges. These are those bridges. Here you can see the corpus callosum. And it's inside this longitudinal fissure. And thousands and thousands and thousands of axons run through here. And there you can see the corpus callosum and these bundles of axons. So just tons and tons and tons of axons run through here. And so you have corresponding areas on each side of the brain talking to each other. Again, you can see it here. Here the corpus callosum is green. Another type of white matter we have are called association fibers. And association fibers are only on one side or the other. They don't cross from side to side. So what they do is they connect different areas within the same hemisphere. So you can see these little blue ones, those are called arcuate. These little red ones, those are called the longitudinal fasciculite. You don't have to know those words, but you do need to know their association fibers. And so when you look at association fibers, you see thousands and thousands and thousands of these things. And it looks like this, all of this. And the reason they have different colors here is just so you can see corresponding areas. But this is what it looks like. So you can see that corpus callosum in the middle, but when it gets out to the sides, it's all over the sides as well. And then there's one other type of this white matter. These are called projection fibers. They're sort of blue in this color picture. So projection fibers, what they do is they connect higher brain to lower brain, or brain to spinal cord. So in other words, they run up and down. And if you look in here, you can see some that are running up and down. If you start at the spinal cord and work your way up, you'll see a V shape. And that V, those are projection fibers. Now it's labeled the cerebral peduncle, and that's the name of that particular projection fiber. But that's what it is. So if you look in this picture, you should be able to pick out commissures, corpus callosum. Those association fibers, they're all spread out everywhere. And then the projection fibers going up and down. You can also, in this picture, you can see the ventricles. So those black spots in the middle, those are those hollow spaces we talked about earlier. And remember, they have cerebrospinal fluid in them. And so you can see the two lateral ventricles, you can see the third ventricle, you can see the cerebral aqueduct, which is sometimes called mesencephalic aqueduct. You cannot see the fourth ventricle in this picture. So that's the white matter. The last thing we're going to talk about today are those little islands of gray matter. So the little islands of gray matter are called cerebral nuclei. Sometimes they're called basal ganglia. But they're little masses of gray matter found within the white matter. And so they look like little islands. Well, those islands are actually all connected to each other. And they're connected to each other, and it looks something like this. It looks like a little animal curled up in each side of the brain. The whole little animal, if you want to call it that, is called the corpus striatum. Corpus means body, and striatum means striped. So this purple thing on both sides is this corpus striatum. But look at this thing. It looks like a little animal, but it looks like it has a head. And it looks like it's got a long tail. And it looks like it's got a little bubble on the end of its tail. Well, each of these is a different cerebral nucleus or basal ganglia. Here it is again. You can see this little animal curled up. And remember, it's on both sides. Well, if you make a section through it, though, it's not going to look like a little animal. You're just going to see parts of it. And so when you make these sections, you can see them here. This is a frontal section. And if you look down at the frontal section, you'll see the pink and the purple. That's the parts that we're able to see. The pink, the red, the purple. So the pink part is like the head. The red part is like the tail, and the blue part is that little bubble on the end of the tail. So what we have are different nuclei. The head part is called the lentiform nucleus. The tail part is called the caudate nucleus. And even though it's not a basal nucleus, the little bubble is called the amygdala or amygdaloid body. Also, though, if you look at this picture and look at the head, you can see that the lentiform is actually composed of two smaller nuclei. One's called the putamen, and one's called the globus pallidus. So here you can see this gray matter inside. That's that corpus striatum. Again, it's divided up into different parts. So we have the lentiform, that's the thing that looks like the head, and again, it's composed of the putamen and the globus pallidus. And then we have that long, skinny tail, that's the caudate, and then the little bubble on the end is the amygdala. So what do these nuclei do? Well, remember they're gray matter. So gray matter is cell bodies and synapses and dendrites. So we're going to get EPSPs and IPSPs and presynaptic facilitation, presynaptic inhibition. We're going to have those different circuits in here and so on. So they act a lot like the cortex, but we're not conscious of what goes on in these basal ganglia, these basal nuclei. We're not conscious of it. It's subconscious. And so what it does is a little bit hard to describe and a little bit hard to quantify, but we know it does some things. So if you think about very, very stereotypical movements that almost everybody has, these are involved in that. So if you think about what happens to your arms when you walk, they don't stay hanging limp at your side. They move, but they don't move randomly. They move in a very stereotypical fashion. They move back and forth opposite to your legs. Well, in four-legged animals, that's a really important thing. And even in us, it helps keep us balanced. That happens here. And so muscle activity is influenced. We prevent unnecessary or unwanted movements. We have these stereotypical movements. It also regulates the intensity of those movements. That's where we're going to stop for today. Anybody have any questions?